Hello, my name is Peter Karagiannis and I work at the Center for iPS Cell Research and Application. Today I will introduce a special exhibition about iPS cell research at the Kyoto University Museum, entitled The Journey of iPS Cell Research, the World of CiRA. This exhibit is part of the events organized in recognition of CiRA's 10th anniversary and provides a history of iPS cells along with information about iPS cell experiments. First, I would like to begin with some background on iPS cells, whose full name is induced pluripotent stem cells. In general, pluripotent stem cells, including iPS cells, have two attractive features. First, they can be expanded indefinitely, meaning one iPS cell can be grown into countless more. Second, they can differentiate into any cell type, meaning an iPS cell can be converted into different types of cells. What makes iPS cells unique is that they can be prepared from cells in your body. I should stress that cells in the body do not naturally have these properties. However, that changes if they are reprogrammed into iPS cells. This is a microscopic image of iPS cells made from human fibroblast cells. iPS cells aggregate to form the round shape in the middle. Those speckles you see inside the shape are actually iPS cells. A distinctive morphology of iPS cells is their round shape. The elongated cells you see around the aggregate are feeder cells, which are used to nourish the iPS cells. These iPS cells were made from the original reprogramming method, but since then many methods have been used to reprogram iPS cells. iPS cells are expected to contribute to various scientific disciplines. Examples include cell therapies and regenerative medicine, but many more applications are foreseen. Moreover, we expect new fields of life sciences and medical applications to come from these cells. Researchers are using iPS cells to explore the control of cell function and fate through synthetic RNA. They are also using iPS cells to understand the events that determine cell fate. From here, I would like to speak more about CiRA. The general public does not have access to the laboratories. This illustration is intended to give you an idea of the environment in which our staff work. The research environment is key for good science. One feature about CiRA is its open laboratories. This design encourages researchers from different laboratories to interact. To be a good scientist, one must have a deep understanding of his or her own field of specialty. But it is also important to be exposed to ideas outside that field. Along with open laboratories, there is a common space where researchers can share ideas. We also have photos with names of all our staff posted in the inside of the Institute to facilitate interactions. Here we have an illustration of a culture cell room. Cells are susceptible to viral and bacterial infection, so some experiments must be done in special rooms. In the culture cell room, experiments are done on a clean bench or safety cabinet. Other experiments done in areas separate from the open laboratories are animal experiments and drug screenings. So too are the preparation of cells for clinical use. Responsibility for the clinical cell preparation has recently been shifted to the CiRA Foundation. This photo shows some standard tools used for iPS cell experiments. In particular, you see 3 micropipettes. It would not be an exaggeration to say that there are thousands of micropipettes at CiRA. How important are pipettes to iPS cell research? Shinya Yamanaka asked for one to be included in the exhibit about his award at the Nobel Prize Museum. Pipettes are used to transfer small amounts of liquid, less than 1 milliliter and sometimes as little as 1 microliter. For experiments at CiRA, such small volumes are quite regular. Technically, pipettes do not need any special training, and just about everyone at CiRA uses them. Considering the size of a cell, microscopes are also indispensable tools for experiments. This is a general light microscope. Unlike the ones seen in elementary schools, this microscope is inverted because the samples include culture media. We had planned to set up a microscope to observe iPS and other cells in the exhibit, but this plan was cancelled due to COVID-19. This photo shows iPS cells minus the culture media. In media, the cells will adhere to the bottom of the dish and grow and expand. The pink in this photo is the result of staining the cells with alkaline phosphatase. The individual speckles show individual iPS cells. In this video, we see fibroblasts reprogramming to iPS cells. The black spot forming in the middle of the video are iPS cells aggregating. As I mentioned earlier, iPS cells have a tendency to aggregate. Around the iPS cells are feeder cells. A new method that does not use feeder cells has also been developed because the use of feeder cells prohibits the clinical application of iPS cells. Such a method can be seen here. In this case, it's blood cells that are reprogramming to iPS cells. You can see the colony forming in the bottom right as iPS cells aggregate. By changing the culture medium, we can then induce iPS cells to differentiate into other types of cells. In this picture, we see iPS cells that have differentiated into heart cells. These cells beat just like normal heart cells do. Finally, in this video, we see immune cells attacking cancer cells. The red cells are lymphocytes made from iPS cells, whereas the green cells are cancer cells being attacked by the lymphocytes. You can see, once the lymphocytes contact the cancer cells are destroyed. This is typical of the events that happen in the body. Cancer grows when the cancer cells evade or resist lymphocytes. One new strategy for cancer therapy being explored at CiRA is the transplantation of iPS cell-derived lymphocytes. Here we return to the exhibit, specifically an experimental room inside CiRA. Users wear a lab white coat during experiments, both to protect their own body from the experimental material and to protect the cells from contamination. This video shows a clean bench. Inside is free of any bacteria or contaminants that could compromise the cells. Another similar system is the safety cabinet, which is commonly used to genetically manipulate cells. To conduct the experiments, scientists open a hatch, but only a little, so that only their hands can enter the clean bench, and only after disinfecting their hands. Similarly, any equipment brought into the clean bench, such as a pipette, is first sterilized with alcohol. After use, the hatch is closed and a UV lamp is turned on to sterilize the inside. A CO2 incubator is used to culture the cells at 37 degrees in the appropriate CO2 concentration. The incubator in this picture was used to culture the first reprogrammed iPS cells. The incubator can be found in the CiRA gallery and has become a popular photo for many of our visitors. I have shown you some of the experimental facilities at CiRA. One point I want to make clear is that everything we have built is not only because of our dedicated staff. Patients, donors, and many others have helped us realize CiRA into what it is today. We had originally planned to ask visitors to write their comments about the future of iPS cell research at the exhibit. Because of COVID-19, we collected comments online from our virtual visitors. We received many messages. In response, we will continue to work diligently in our goal to create new medicine from iPS cells. Thank you for watching.